I'll just start introducing this seminar series. So this is seminars that we started out of the network that we have in France that's called the GDR Plasticité. So it's a network about phenotypic plasticity, and we are organizing normally national meetings, exchange, collaborations, etc., funding a few trips for that. Since last year and the pandemic, we started these online webinars where we invite some people outside France, which is great for us. So this time we have the great pleasure to have David Pfennig, which I will let Vincent Debat introduce. Vincent is co-heading this network with me.
Okay, so hello to everybody. So as Miguel said, it's a great pleasure to have David Pfennig with us today. So as you might know, David's been working on phenotypic plasticity for quite a long time now, and in particular the evolutionary consequences of phenotypic plasticity. He started or he obtained his PhD from Austin University in Texas, working with James Bull and Mark Kirkpatrick. And then he spent most of his professional career at the University of Northern Carolina, where he's pretty strongly involved in teaching evolutionary biology, as I've found on his website. In terms of research, so he's got a pretty nice research record on various topics, including frequency-dependent selection and the evolution of Batesian mimicry in snakes, cannibalism, so this is on spadefoot toad tadpoles, character displacement, the evolution of character displacement, and the evolution via genetic assimilation involving the phenotypic plasticity effect. And so, yeah, today he will be talking about the evolution of plasticity and the consequences on the origin of novelty. So that's a pleasure to have you here today, David.
So, sorry, I forgot to mention something. There's a Q&A question and answer tab in the bottom. You can type your questions there during the talk and at the end, and then you'll be able to ask your question. Also, we'll turn on your microphone, but first, please type your questions there. And people can vote for the questions they like. Sorry.
All right, well, thank you, Luis and Vincent. Thank you for the invitation, and thanks everyone for coming today. It's great to have this opportunity to speak to y'all, and hopefully everybody's doing well. You know, this are obviously crazy times, and it's great to be able to have this opportunity to share some science with you. So let me just go ahead and begin by starting my screen here. So what I want to talk about today is I want to talk about the possible role that phenotypic plasticity might play in the evolutionary origins of novel traits. And let me just begin as background and note that, you know, one of the central challenges that we face in evolutionary biology is to explain where novel features come from. So where do features such as flowers and feathers and eyes ultimately come from? Now, I think it's fair to say that traditionally evolutionary biologists have focused on this issue by examining the genetic and ecological forces that are thought to promote innovation. And in contrast, the role of development, I think, has been relatively understudied. And yet it's becoming increasingly clear that a key feature of development, its capacity to be adaptively responsive to changes in the individual organism's environment, might play a critical role in the origins of novel traits. And it's this topic that I want to focus on in my talk today. So I'll be talking about a phenomenon known as phenotypic plasticity, which I'm sure you're all familiar with. I'm going to use it synonymously with developmental plasticity. And I want to just start off by just defining my terms. So I'm going to define plasticity as the ability of an individual organism to alter some aspect of its phenotype in direct response to a change in its environment. So let's just look at a couple of examples. So sometimes plasticity can produce really profound change from a single genome. So for instance, in response to increasing photoperiod, monkey flower plants will shift from producing the open flower that you see on the left there to producing closed flowers like you see on the right there. But sometimes these effects can be pretty subtle. So for instance, in response to eating more meat, spadefoot toad tadpoles express a peptidase gene more highly. So when you have these really profound examples, like on the left, we refer to these as a polyphenism, where you get this sort of discrete variation. But then the more sort of gradual variation that you see on the right, we refer to as a reaction norm. Now, plasticity is something that evolutionary biologists have known about for a long time, of course, and it's also been a puzzle to evolutionary biologists how plasticity fits within their framework for explaining how living things evolve. And so this is really the central question I want to examine today, which is what is plasticity's role in evolution? So I think it's safe to say that if you were to ask most evolutionary biologists this question, even today, you'd probably get an answer somewhere along the lines of this quote from Meyer from 1963, where Meyer said the ability of the phenotype to respond to the demands of the environment without mutation, that is the ability to express plasticity, greatly reduces selection pressure. So in other words, what Meyer is saying here is that the ability to express plasticity should essentially weaken selection. And so that should act as an impediment force in evolution. Now, opposing this viewpoint that plasticity slows evolution down is another viewpoint which suggests that instead plasticity speeds evolution up. Now, this is an idea that's been around for a long time, actually well over 100 years, but it's been most prominently developed by Mary Jane Weiss-Eberhard in her book, Development, Plasticity, and Evolution. And in this book, she states that most phenotypic evolution begins with environmentally initiated phenotypic change. And then she goes on to say genes are followers, not necessarily leaders in phenotypic evolution. So what I want to do today is I want to examine this latter idea. And I specifically want to ask, well, how is it that plasticity can actually speed evolution up? So an outline of my talk is as follows. We're going to begin by asking how plasticity can lead evolution. We're then going to ask, how do you test these ideas in natural populations? So we're going to examine how you test for plasticity-led evolution in natural populations. I'm then going to focus most of my effort today on describing a case study whereby we can go about testing these ideas in natural populations. And then I want to just end with some general considerations about plasticity-led evolution. So let's begin with asking how plasticity can lead evolution. Now, there's actually several different ways that plasticity can theoretically facilitate evolution. I'm going to focus today on two primary mechanisms. One is an indirect method or an indirect route by which plasticity can affect evolution and speed it up. The other is a more direct way that it can do so. And we'll start with the indirect hypothesis first. And I'm going to refer to this as the buying time hypothesis. And underlying this idea is that because plasticity allows individuals to match their phenotype to current conditions, plasticity is often beneficial. That is, it's often adaptive. And to illustrate this, we can turn to work done on water fleas, Daphnia. And as many of you know, when water fleas are exposed to predators, at least certain lineages, they will sometimes then express this distinctive helmeted form that you see on the right there. Now, if you look at the survival of these two different forms in the presence of predators, you find that whereas the form, the normal form on the left there has less than 10% survival, the form that's induced by the predators, the helmeted form, has greater than 90% survival in the presence of predators. So this is telling us then that this example plasticity is adaptive. Now, because plasticity is often beneficial, that is because it's often adaptive, it should increase a lineage's chance of persisting. So for instance, bird species that are more plastic, as measured by a higher propensity to behavioral innovate, are actually at a lower risk of extinction. Now, this is important because if plasticity does tend to increase a lineage's chances of persisting, then it suggests that plasticity might essentially buy time until a lineage can acquire genetic changes to adapt to rapidly changing environments. And then ultimately, it goes on to diversify. So in this way, plasticity can basically speed evolution up. And consistent with this hypothesis, taxa that have actually evolved conspicuous plasticity, like in this case, this plasticity and resource use, they tend to be more species rich than their sister taxa that have not. So in this particular case, we're looking at examples in which different fish and amphibian lineages have evolved resource polyphenisms, and those clades that contain those resource polyphenisms are more species rich than their sister clades that do not. So again, this is all consistent with this idea that plasticity might essentially facilitate evolution indirectly by just enhancing lineage persistence. Now, I want to just say that in my mind, this is probably the most important facilitatory effect that plasticity can have on evolution. But what we're going to do today is we're going to shift gears and look at a second way that plasticity can affect evolution more directly. And this is what I'm going to refer to as the plasticity-led evolution hypothesis. And to illustrate this idea, I'm going to use a series of drawings, of cartoons. So we're going to begin with a genetically diverse population. And in my case, I'm going to just illustrate these as tadpoles there. And notice that they're all the same shape, meant to signify that they produce the same phenotype initially, but I'm showing them as different colors. And these different colors are meant to signify different genotypes. Now let's imagine that our population experiences a novel environment shown by the shaded background here. Now, exposure to a novel environment is typically stressful to all organisms. And the primary way that organisms deal with the stress associated with a change in their environment is to use their inherent plasticity to produce traits that are a little bit better at dealing with that environment. So what we see here then is that novel traits then are induced by this novel environment. But here's the key point. Different genotypes are likely to respond differently to the same change in the environment. And indeed, in almost every natural population where this has been studied, we find that different genotypes differ in whether and how they respond to any given change in the environment. So in our case, you can see that the brown genotype changes by producing a narrower bodied tadpole, the blue genotype by producing a fatter bodied tadpole, and then the green genotype is in between those two. And then lastly, the yellow genotype is not changing at all. So in essence, what's happened here is that there's cryptic genetic variation that's been revealed by this change in the environment through the plasticity. Now, once that variation then is revealed to selection, then selection can act on that variation, and it can start to disfavor the maladaptive traits and hence the maladaptive genotypes in this new environment. So let's imagine that the blue shape, the blue genotype that produces that fat bodied form, for whatever reason, let's imagine that form is now favored in this novel environment. So selection can basically favor that genotype, that form, and basically eradicate all others from the population. But it can also start to refine that favored trait. And I'm showing that by the blue tadpole being a little bit larger. Now, this selection might eventually result in a situation where everybody in that novel environment then begins to produce this new phenotype, right? So they produce this fat-bodied form in the novel environment, and then they produce the narrower-bodied form in the ancestral environment. And if you have a situation like that, where you get discrete phenotypes being produced in response to environments and different situations, then you have what's called a polyphenism, as we saw earlier with the monkey flower plant. But selection might actually be so powerful or so effective that it might eventually cause a formally induced trait to become fixed in the population. So even if our population now experiences the ancestral environment, then they may have no options other than to produce now that fat-bodied form. It might essentially become canalized or fixed in the population. And if that happens, then we have the process of genetic assimilation occurring. Now, the important point I want to make here is that regardless of the outcome being a new polyphenism or a new fixed trait as a result of genetic assimilation, in both cases, you end up with a new novel complex trait through this process. And I want to emphasize that really what happened in this process is that it was the plasticity that basically started this process in motion by revealing that variation to selection that selection was then able to act upon. So what have been the reaction to these ideas? Well, not surprisingly, there's been a fair bit of skepticism. And I think the skepticism is best captured by this quote from Doug Fatuma. And I use this quote from Fatuma because he's one of our big thinkers in the field. He's publisher of one of our primary textbooks on evolution. But also he's somebody who thinks a lot about these problems. And when he wrote this review in 2015, he said considerably more research on a range of taxa and characters will be needed before we can judge whether or not plasticity often leads adaptation. So that was really the charge, I think, for those of us who work in this field, is that we really needed to go out and gather more data and really find ways to test these ideas in diverse kinds of settings. And so that's what I want to talk about next. I want to ask, well, how do we test for plasticity-led evolution? So a few years ago, I got together with one of my now former graduate students, Nick Levis, and we decided to come up with a way you can test these ideas and again, in natural populations. And we began our project by basically noting that, you know, these ideas are controversial primarily because comprehensive tests for natural populations are generally lacking. And I want to just say quickly as an aside, you know, at the time we referred to this as the plasticity first hypothesis or plasticity first evolution. For reasons which I'd be glad to get into afterwards. I think referring to it as plasticity-led evolution is probably a little bit more accurate. But anyway, so what we set out to do in this review, which we published in Tree, was to basically come up with a means by which the role of plasticity and adaptive evolution could be assessed, right? And so this is really the idea that we set out to do here. And so one of the things that we tried to do was to come up with key criteria or key predictions that you could apply to any given system and use these criteria to evaluate whether or not plasticity-led evolution can account for the occurrence of some novel complex trait that you're focusing on in that particular system. And so we came up with 4 key criteria, and I'm going to go through each of these individually. So the first and probably the most fundamental of our criteria was that the focal phenotype, or more likely its components, will be environmentally induced when ancestral lineages experience the novel, that is the derived environment, or the environment that's normally associated with that novel complex phenotype. So basically what we're saying here is that there should be, of course, some evidence that as you work your way backwards through a phylogeny, if you want to think about it that way, you should find some evidence that there was pre-existing or ancestral plasticity. The second prediction is that cryptic genetic variation will be uncovered when the ancestral lineages experience the novel environment. And this criterion is important because remember, it's the uncovering of that cryptic genetic variation which basically fuels this process of plasticity-led evolution. And then thirdly, there should be some evidence that there's actually been evolution. So the focal phenotype should show evidence of an evolutionary change either in regulation, that is in its degree of plasticity, and or in its form in the derived lineages. And then lastly, there should be some evidence that the focal phenotype has undergone adaptive refinement in the derived lineages. So in other words, through evolutionary time. And the reason this criterion is important is because remember, it's selection that's driving this process. So you really want to see if there's a signature of that selection in your particular system. So notice that in evaluating these criterion, you're going to be faced with comparing derived lineages that contain the novel complex trait that you're interested in studying and what we're referring to as ancestral lineages. Now, of course, if you're doing experimental evolution studies, like some of you are doing, I'm sure, then you can obviously make that comparison very directly. But what we were setting out to do in this analysis was to come up with ways that you could evaluate these criteria in natural populations where you wouldn't have access to this sort of really cool experimental evolution studies. And there are really 2 main contexts, I think, for comparing ancestral and derived lineages in natural populations. And one way or one context to do this is to take advantage of range expansion studies. So for instance, if you have a situation where your species of interest has undergone a range expansion, then the source population can basically act as your ancestral population, and then the sink population, or the population that the disperser you're moving into, they can serve as your derived populations. You can basically compare those contemporary populations simultaneously. But another way you can do this is by using what we refer to as ancestor proxy studies. So what I'm showing you here is an ancestral character state reconstruction, and I'm showing you a derived lineage out there and then the ancestral lineage back there. And I'm mapping on this some character state. And so, more ancestral being cooler colors and derived being the warmer colors here. And so let's imagine that your complex trait that you're trying to look at is in that derived lineage there in the dark, the red coloration. And what you want to study then, of course, is that ancestor. But the problem obviously is the ancestor is extinct, so you can't refer to that ancestor. But what you can do is you can use an extant species that you infer has the same character state as the ancestor had, but that's still with us. And then you could basically use that ancestor proxy to compare to the derived lineage. And so in the case study that I'm about to go through here in just a moment, you're going to see that we're going to use both these ancestor proxy studies as well as range expansion studies to evaluate the 4 criteria that I laid out a moment ago. So we are going to turn now to our case study in natural populations. And let me just preview this section by just saying that I don't want to imply that the particular case study that I'm going to go through here is the exemplar of plasticity-led evolution. I'm sure it's not. I simply want to use this example to illustrate how you could go about evaluating these criteria in natural populations. So the organisms that I'll be talking about today are these guys, spadefoot toads. I'm going to spend a little bit of time talking about spadefoot just so you can understand the natural history of the system that we're dealing with here. So these are the New World spadefoot toads that you see here. There's 7 species. They belong to 2 different genera, the genus Spia, which has 4 species, and then the genus Scaphiopus, which has 3 species. Now you can see they're found throughout North America, from the Atlantic to the Pacific, Canada to Mexico. But you can see that they really reach their highest diversity down in the southwestern deserts of the US and northern Mexico. And so I want to tell you a little bit about their natural history. So these guys are called spadefoots because they've got this keratinized structure on the back of their feet called a spade. And they use that to dig into the ground. And the reason it's important for them to dig underground is because they live in environments like this. This is a picture of our field sites in southeastern Arizona, southwestern New Mexico, and this is typical spadefoot habitat. So this doesn't look like a good place for amphibians to live, right? So they're actually spending most of the year deep underground, encased in a mucus cocoon in a state of estivation. But like all amphibians, they have to have water in which to breed. And in the American Southwest, this water comes in the form of these brief, heavy summer rainstorms. So these rainstorms induce the toads to come above ground. The rainstorms also cause water to form in low-lying areas. And it's within these low-lying areas that our story today will be taking place. And so the adults will congregate around these temporary pools. The males will call to attract the females, and then all breeding will take place in a single night. The eggs hatch in about a day to 2 days, depending upon the species. And then the tadpoles are in a race to get out of this pond before the pond disappears, before it dries up. So here you see a little toadlet, a tadpole that's transformed into a toad, emerging on the land. And those other sort of blobs in the background there are other tadpoles, which may not make it out of this drying pond before it completely disappears. So if we go into one of these ponds and we look at the tadpoles, we find something really interesting. So some of the tadpoles in these ponds look like tadpoles you would see anywhere in the world. So the tadpoles that you see on the margins of this image here. So they're pretty standard looking tadpoles, but then you see other tadpoles in the pond that are very strange looking, like the guy there in the middle. And so they're much larger, they've got this big head, and also behaviorally they're different. So they look like they're very aggressive, they're very active, moving around the ponds. And so what we have here is we have a polyphenism. We have some tadpoles that look like the form on the left there, and we have other tadpoles that look like the form on the right there. Now, if you look at these in more detail, you find that they differ in a number of important respects. So the tadpoles, the smaller tadpoles that look like the one on the left, have a really long gut. And so you can see this photo of the gut. You can take a little tadpole that, is the size of smaller than your thumb, and its gut might be 1 meter long. Because it's coiled back on itself. Whereas the other tadpole, this large-headed form, has a really short gut. So it's about 1/5 the total length of the smaller tadpole's gut. If you look at these large-headed forms front on, you find that they also have these enlarged serrated beaks made out of keratin. Sort of looks like a hawk or an eagle's beak. But also what's important here is that is that these essentially represent, like I said, two different morphs. And so we're going to refer to these guys as on the left as the omnivore morph, and I'm going to refer to the ones that look like the form on the right as the carnivore morph. So this is a polyphenism, as you'll see in a moment. Diet plays an important role in influencing whether an individual develops into one form or the other. They're essentially born as omnivores in most cases. And then if they eat meat, like these anastracan furry shrimp, which you see there in the middle, then at least some individuals have the potential to go on to produce that carnivore morph. And I can illustrate that by showing you the results of a simple experiment. So what we did in this experiment is we took different families, different SIB ships. These are Spia multiplicata families, and we split them up into two different groups, and we fed them either on detritus exclusively or on shrimp. And then after about a week and a half, we measured their overall morphology. And so on the left there, on the y-axis, you can see this morphological index. So this is a composite shape variable. It's basically a principal component score that takes into account various component traits that comprise that the overall variation between the carnivores and omnivores. So for our purposes, then more positive values or higher values are more carnivore-like, lower values are more omnivore-like. So what you can see here, first of all, is that you can see that carnivores are only produced when individuals eat meat, and they have to eat that meat early in life in order for the carnivore to be triggered. But what you can also see here by comparing the different lines here, and those different colors represent different families, is that different families or different SIB ships / different genotypes differ in their propensity to produce carnivores. So just as I was talking about earlier, it's this underlying heritable variation that basically is going to fuel the evolutionary responses we're going to be looking at here for the next several minutes. So a couple other points I want to make about this system. So one is, as their names imply, these two different forms feed on different, basically occupy different ecological niches. So the omnivore is a dietary generalist. They eat plants, detritus, and small crustaceans. The carnivore morph, in contrast, is a dietary specialist that preys mostly on these large anastracan furry shrimp, which are actually one of the important cues that induces their production in the first place. They also will feed on other tadpoles, so they're cannibalistic. Now, one last thing I want to explore with this system is just to ask, well, why do we have a polyphenism here? And so that's an important question to ask because, you know, sort of our caricature of how selection should act is that selection should favor some optimal trait value in a population, and it should tend to eradicate other extreme variation around that optimal trait value. But of course, that sort of stabilizing selection isn't the only way in which selection can work. And it can also operate in a disruptive fashion, where the extreme traits are favored and intermediates then are disfavored. And so one of the questions we set out to ask is, well, is disruptive selection acting in this system and presumably other systems like it? And can that explain the maintenance of this polyphenism? And so I'm showing you the data here from one pond, but we've since done this in now dozens of ponds and find essentially the same thing. And so again, on the x-axis, as you saw earlier, this is a morphological index, so more omnivore-like to the left, more carnivore-like morphology to the right. And then the y-axis here is a proxy for fitness. And our proxy for fitness here is to use body size or snout vent length. And snout vent length is a good proxy for fitness here because larger tadpoles have higher survival. They reach metamorphosis at larger size. Individuals that are large at metamorphosis mature as larger adults. Larger adults have higher mating success and higher reproductive success. So what you can see here is each dot in this graph is an individual tadpole. So one point I want to make here is that you can see that intermediates actually are produced early in development. But the important point is that those intermediates appear to be disfavored. So they have lower fitness than the extreme traits do in this population. And we think the reasons for that is that essentially the intermediates are not well adapted for either of the main resources that are available in this system. So omnivores are really good at utilizing detritus and plant material because they've got features like this really long gut that allows them to process that resource. Carnivores, on the other hand, have these large jaw muscles and the keratinized beak, and that allows them to utilize the mobile large active prey like the furry shrimp. And then intermediates are just lousy at both those two. They're not as good or as effective at utilizing either of those two resources. So that explains how you get the disruptive selection. But we have one other question that we need to answer, which is why is it that one of these morphs doesn't eventually supplant the other in the population? That is, why do we still get the maintenance of the polyphenism? And the answer to that question seems to be that we have operating also in this system inverse frequency dependent selection. That is, that individuals are favored when they're rare, but as they become more common in a population, selection disfavors them. And we can illustrate this by doing the following kind of experiment where we take a pond and we divide it up into different sections, and then we redistribute the tadpoles such that we increase the carnivores in some sections or decrease the frequency of carnivores in other sections, increasing the frequency of omnivores, of course, in those latter sections. And then what you see is over time, in a fairly short period of time, the different sections, the different treatments converge around a common equilibrium value. In this particular pond, that equilibrium value was about 50%, but that value varies from pond to pond depending upon the relative abundance of the shrimp and the detritus in the pond. But the important point here is that what this is telling us then is that neither morph is intrinsically superior to the other. And so because of this frequency dependent selection, we get the maintenance of these two morphs. That is, we get the maintenance of polyphenism. And again, we think similar processes probably operate in many other systems where you have polyphenisms. So having looked at the natural history of the study system, what I want to now do is spend the remaining time applying these criteria that I outlined earlier to evaluating the plasticity-led evolution hypothesis in spadefoots. And the specific feature that we're trying to explain is this novel complex carnivore morph. And you'll see in a moment that there is evidence that it is indeed a novel trait. And so this is just to remind you what those criteria are. Again, I'm not going to go through these individually because we're going to go through these in the next several slides. But we're going to apply these criteria again to the spadefoot system. So we have this novel polyphenism where we have carnivores and omnivores. And essentially what we want to do is we want to work backwards through a phylogeny, and we want to find an ancestral lineage that we can utilize to ask, is there any evidence for any sort of pre-existing or ancestral plasticity in the system that might have sort of presaged or might have initiated the evolution of this novel polyphenism in the system. So here's an ancestral character reconstruction of the New World spadefoots, and then as the outgroup, there are other families of frogs, so Pelodiotidae, Megaphrynidae, and Pelobatidae. And what you can see on this phylogeny is I've shown the tadpole phenotype. The blue letters are the observed tadpole phenotype. So the O's mean they produce only omnivores. The B means they produce both morphs, that is, they have the polyphenism. And then the C, there's that one lineage in the middle that I'll come to in a moment where they appear to be fixed mostly for producing only carnivores. So one thing you can see from this phylogeny is that this is a novel trait, right? So it's restricted to just this one genus and the members and the genus Spia. So what we want to do is we want to look at the origins of that novel polyphenism. And we want to work our way back to the ancestor of Spia before that polyphenism evolved, indicated by the red arrow there. And we inferred that that ancestor must have only produced omnivores, since that seems to be a character state for frogs generally. But of course, we have the problem of that ancestor is extinct, so we can't access it. But what we can do is we can use members of the modern genus Scaphiopus to serve as a proxy for that ancestor. And so we're going to use this ancestor proxy approach. Now, Scaphiopus serves as a proxy for the ancestor of Spia before the evolution of carnivores for three reasons. One is it's the sister genus to Spia, as you can see on this phylogeny. Secondly, they're ecologically similar to Spia. In fact, they breed in a lot of the same ponds. They experience a lot of the same conditions. And then lastly, and importantly, Scaphiopus, as you can see, does not express the polyphenism. And again, as I said a moment ago, the lack of that polyphenism or just producing omnivores is the inferred state for the ancestor and for anurans, that is frogs and toads generally. So we begin by using Scaphiopus as our proxy for the ancestor to Spia. And we begin by testing our first criterion. And as a reminder, our first criterion was to ask, do novel environments trigger pre-existing plasticity in that ancestral lineage? So for the purposes of this slide, I'm going to focus on Scaphiopus holbrooki as our ancestor proxy. And what we're going to do here is just like in the slide I showed you earlier, we're going to basically take different SIB-ships, different genotypes, and we're going to rear them either on their ancestral diet or their standard diet of detritus, or we're going to force them to eat this novel diet of shrimp. So this is not something they would normally eat in the wild. And what I'm showing you here then is for combining all these SIB-ships together, we're looking at the trait values that they produce on detritus on shrimp for 4 different component traits that basically constitute that complex carnivore phenotype. So a number of identical rows, mouth part score, jaw muscle width, and gut length reading from top to bottom. So the asterisks indicate traits where the differences between the diets were significant. And what you can see is that for 3 of our 4 traits, the only exception being jaw muscle width, they produced a different trait value on shrimp than on detritus. So I want to emphasize here that, this is telling us that the ancestor proxy exhibited plasticity in 3 of our 4 traits, but it's not conspicuous plasticity. I mean, here's a Holbrooki tadpole, and you can see he's eating on another tadpole. And his gut length is not super short. It's not super impressive. It's nowhere near the shortening of the gut that you see on a spia carnivore where they have that polyphenism. But nevertheless, this is telling us that there does appear to be some semblance of plasticity, diet-induced plasticity, in these component traits that constitute the carnivore phenotype. So we then take it that our first prediction, or we conclude that our first criterion is satisfied. So our second criterion was to ask, do novel environments uncover cryptic genetic variation in our ancestor proxy when they experience the novel environment? So for these data I'm going to show you, we're going to use a different ancestor proxy. Now we're using Scaphiopus couchi. And again, same experiment, feed some SIB-ships, members of some SIB-ships on detritus, some on shrimp. And then these are the reaction norms for the 8 different genotypes / SIB-ships that we use for this experiment. And what you can see is there's a lot of convergence in terms of gut length, which is what we're looking at here, when they're fed on detritus, which is their standard diet. But you can see that there's a lot of spread among these 8 genotypes and gut lengths when you look at what's happening on the shrimp diet. Now, that's suggestive that, in fact, cryptic genetic variation is being uncovered on the novel environment, but we can quantify that cryptic genetic variation by basically using heritability as a proxy for that cryptic genetic variation. And not surprisingly, we get no significant heritability on detritus, but we do get significant heritability on shrimp. So what this is telling us then is that the novel environment appears to have uncovered cryptic genetic variation in our ancestor proxy. So we conclude that our second criterion is satisfied. So our third criterion was to ask, is there any evidence that our novel trait, once it's been revealed in that ancestor proxy, that it underwent evolution? And specifically, has it evolved in regulation and or in form? So I'm going to spend a little more time on this criterion because there's a little bit more involved here. And so we've seen up until now, we have this novel polyphenism. We've worked our way back using this ancestor proxy approach, and we found evidence that that novel polyphenism may have been preceded by ancestral plasticity or by diet-induced plasticity of some type, in our ancestor proxy. So what we want to do now is we want to focus on this lineage in the middle, that Spia bomifrons sympatric lineage. And I'll explain what I mean by the sympatric here in just a moment, because that lineage appears to have evolved a situation where they are only now reproducing that carnivore morph. So we have this third lineage we can draw into here, where our phenotype has undergone canalization or near canalization. And so what's happening here is that these bomifrons that are in sympatry, they refer to sympatry with another species of Spia, Spia multiplicata. So these are the ranges, geographical ranges of Spia bomifrons shown in blue and green, and then the range of Spia multiplicata shown in yellow and green. And you can see there's this area of green in the US states of New Mexico and Texas and parts of Arizona and Colorado, where these 2 species potentially co-occur. Now, we have a number of lines of evidence to suggest that what's happened here is that this sympatric zone represents secondary contact. And specifically, it looks as though Bomafrons has very rapidly and recently expanded its range out of its normal range in the Great Plains of the US down into the deserts of the Southwest. There's a whole interesting story I'll be glad to get into afterwards about what might be going on there with regard to what's driving that range expansion. But for our purposes, what this means then, if that is indeed secondary contact, then is that our allopatric populations of each of these two species represent the ancestral condition, whereas the sympatric populations then represent the derived condition. So here we're going to use range expansions as a way of testing ancestral and derived lineages, as we outlined earlier. So if we look at our, because remember the carnivore phenotype is what we're interested in explaining here. And if we look at the production of carnivores in each of these two species when they're in allopatry, we find that they don't differ. They produce similar intermediate frequencies of carnivores and omnivores when they're found in allopatry. And remember, that we infer is the ancestral condition. But if we look at what's happening in sympatry, the derived condition, we find that in Spia multiplicata, they produce mostly omnivores, very few carnivores, whereas Spia bomafrons is producing mostly carnivores, very few omnivores. So what's happened here is that these two species have undergone character displacement. That is, selection acting to minimize competition for resources has basically caused them to evolve greater differences here. So those differences then we can look at in more detail by focusing now on Spia bomafrons, and taking these adults from these populations, rearing their offspring in the lab under common conditions. And what we find is that the animals whose parents were derived from sympatry are actually more carnivore-like at birth. So these are 2-day-old pre-feeding tadpoles. They have not experienced the cue that normally induces the carnivore morph. And you can see they're producing more carnivore-like tadpoles just from birth. So that's suggesting that they have in fact evolved in regulation. They're becoming less plastic, if you want to think about it that way. But we can also look at this at the sort of molecular level. So we can look at molecular phenotypes. And it's particularly what we're doing is we're looking at divergence between these allopatric and sympatric populations of bomafrons that are undergoing this character displacement in their gene expression profiles. So these are combined gene expression profiles for about a dozen candidate genes that are differentially expressed between the two different morphs. And on the left panel is where the parents were derived from allopatry, and on the right is where the parents were derived from sympatry. And then the blue ovals is showing you the combined gene expression when those tadpoles were reared on detritus, and then the yellow when they were reared on shrimp. And what you can see here is, just as we see with the morphology, the sympatric tadpoles have lost the diet sensitivity, in this case, in gene expression that was present in the allopatric tadpoles. So the bottom line here is that it looks like the phenotype has evolved in degree of plasticity. They're becoming less sensitive to the environment. They're becoming basically moving towards fixation of only producing the carnivore morph. In other words, they've evolved in regulation. But the phenotype also appears they've evolved in form. And one of the things we discovered as we were studying, as we were conducting this study, is that when we looked at bomafrons carnivores, we found this novel trait that had not previously been reported. And so what this trait is we refer to this as a palate spike. And so what you're looking at in that photo in the middle there with the yellow arrow is you're looking down inside the mouth of a bomafrons carnivore, and you see this keratinized spike, this structure coming out of the top of the mouth. We don't know for sure what that structure does. We suspect that it probably is used to impale prey and hold them in place while they feed on them. But the important point is that this palate spike is a novel feature that's restricted only to Spia carnivores. So the bottom line for this purpose, for our purposes here then, is that the phenotype has not only evolved in regulation, but it's also evolved in form. So we conclude that our third criterion has been satisfied. Now our fourth criterion was to ask, has the novel phenotype, our carnivore morph, has it undergone adaptive refinement? And so I'm going to start this part off by showing you data that you saw earlier. So these are those component traits of the carnivore morph from the ancestor proxy from the Scaphos holbrooki when they're reared either on detritus or shrimp. And now what I've done on each of these panels is I've overlaid on top of those panels a gray arrow. And that arrow is indicating the direction of the adaptive change that we expect the plasticity to exhibit, if the plasticity were adaptive. So for instance, number of identical rows, we expect them to produce more identical rows when they're fed on detritus than when they're fed on shrimp, because these identical rows are used for rasping material off the bottom of the pond or off rocks and stuff like that. Whereas for mouth part scores, we'd expect them to have higher values of that when they're fed on shrimp than on detritus, because that means that the mouth part's being better developed for grabbing onto the shrimp, and so forth. So what you can see is that whereas some of these traits are in fact moving in the adaptive direction, some of them, like the mouth part score, are not, right? And so there's pre-existing plasticity in this ancestor proxy, but it's not necessarily adaptive, and it's not necessarily coordinated. Now you compare that situation to what you see in a species that has evolved the polyphenism, so in this case, Spia multiplicata, and you can see that the plasticity is going in the adaptive direction for all 4 of these component traits. So unlike the ancestor proxy, Spia multiplicata, the derived species that has evolved the polyphenism, exhibits adaptive plasticity in all traits. But also we can look at how well they grow on these different diets, on the detritus and shrimp. And you can see that our ancestor proxy actually performs really poorly on the shrimp. In fact, that some of them actually will starve rather than eat those shrimp, because it's, it's like feeding you cardboard. You wouldn't recognize it as something to eat. But in contrast, unlike the ancestor proxy, Spia multiplicata grows equally well on both diets. And so this indicates, again, we take as evidence for adaptive refinement on that novel diet. But there's another way we can test this criterion about whether our phenotype has undergone adaptive refinement. And that is to evaluate this prediction, which I think really underlies this whole plasticity-led evolution hypothesis, which is that the more frequently a novel trait is expressed, say through plasticity, the greater the magnitude of its adaptive refinement. And so we can apply that prediction to our system by, again, coming back to our scenario where we have the character spacing occurring between bomifrons and multiplicata. And so again, if we focus on bomifrons, these populations in sympatry versus allopatry, in sympatry, the carnivores are being expressed frequently, and so they would presumably undergo greater adaptive refinement in those populations. Whereas in allopatry, the carnivores are normally expressed much less frequently, and so they have less opportunity to undergo adaptive refinement. So another way of thinking about this is that if you collect parents from these 2 different regions, from sympatry and allopatry, and you take their tadpoles, and you essentially compete them against each other from those 2 source populations, but compete them against each other either when they're competing for detritus versus shrimp. On the detritus diet, we expect the allopatric tadpoles to win out, because they're normally utilizing the detritus more frequently because they're expressing the omnivore morph more frequently. But when they're competing for the shrimp diet, you would expect the sympatric tadpoles to outperform the allopatric tadpoles, because again, that carnivore morph, which is specializing for utilizing the shrimp, has undergone, is expressed more frequently, so presumably it would have great opportunity for adaptive refinement. So when we test this idea, we find support for that prediction. So what you're seeing here is when we perform contests between pairs of tadpoles, either for detritus or shrimp, and then the brown bars are when they were when the parents were from allopatry, and the purple is when they're from sympatry. And then the height of that bar is indicating the number of times a tadpole for each of those 2 selective environments won in a contest. And what you can see is that when they're competing for detritus, just as we predicted, the sympatric tadpoles do better. They outcompete the allopatric tadpoles outcompete sympatric tadpoles for detritus. But then it's the opposite on shrimp. It's the sympatric tadpoles that are outcompeting the allopatric tadpoles. Now we think the reason why sympatric tadpoles outcompete allopatric tadpoles for shrimp is because sympatric tadpoles appear to have evolved to eat shrimp faster. And so this is just from birth. I mean, again, these are all reared under common conditions in the lab. And what we're looking at here is shrimp eating times, how long it takes them to eat a standard amount of shrimp. And you can see these sympatric tadpoles are just a lot more effective at getting the shrimp Q, which is the Q that induces that carnivore morph in the first place as well. So you can see that we have multiple lines of evidence suggesting that this novel phenotype has indeed undergone adaptive refinement. So we conclude that our 4th criterion has been satisfied. So to wrap this part of the talk up, in terms of what we think went on in this system, we think that ancestrally, we had a situation where the tadpoles were like modern day Scaphiopus, or like most tadpoles, where they were mostly utilizing detritus or plant material. But they faced a problem, and that is that their habitat, the southwestern US and northern Mexico, we know that for the last 11,000 years has been drying out very rapidly. And so there was a premium on speeding up development to get out of these ponds before they dried up. And one way to speed up development for tadpoles is, of course, to get a more nutritious food resource. And sitting out there was this abundant ecological opportunity in the form of these anastracan fairy shrimp, which the tadpoles could utilize, again, to enhance their development. So presumably a few individuals began to utilize these shrimp. That then increased their chances of getting out of the ponds. And so they took these behavioral tendencies to eat the shrimp with them into the next generation. And then eventually selection started to refine the other features that constitute this carnivore morph that allowed them to be even more effective at utilizing those shrimp. So features like the keratinized beaks, the large jaw muscles, and the short gut. And then in many lineages, you ended up with a polyphenism. You ended up with a situation where you had this developmental switch where they produce either omnivores or carnivores. And that switch or that polyphenism is being maintained in those populations because of inverse frequency dependent disruptive selection, as we saw earlier. But at least one lineage, the Balmofrons that occurs in sympatry with Multiplicata, we've seen that carnivore morph evolving towards fixation. And so we have here a situation where we think we have plasticity-led evolution that can explain the origins of this novel complex feature found in the genus Spia. So I want to just end and spend a few moments by talking about some general considerations that we want to maybe take into account here. One point I want to make here is that plasticity-led evolution may often be initiated by a change in behavior. And so if we go back to the scenario that I just outlined a moment ago, notice that really the key step here was that a few individuals, I said, presumably started to utilize those shrimp. And so it was really that behavior, that tendency to start to eat the shrimp that we think got this whole process rolling and then exposed the cryptic gene variation, presumably to selection and allowed it to undergo that refinement. And this is consistent with what Meyer said, where he said a shift into a new niche or an adaptive zone is almost without exception initiated by a change in behavior. And so when I'm referring to behavior here, I'm not just talking about the sort of, familiar forms of behavior that we see in many animals, but we can also think more broadly about behavior in plants, like the development of root systems, the evolution or the development of the cytoskeletal structure, or even neural development, So all these are examples of sort of exploratory behaviors where populations or lineages can be exposed to new selective environments, and those might play a critical role in initiating plasticity-led evolution in many situations. Another point I want to make here is that plasticity-led evolution can actually promote really rapid change. And to illustrate this, let's go back to our character displacement story in the spadefoots. So we can simulate how that character displacement might have occurred by doing the following experiment in the lab. So we can take Multiplicata and Balmofrons, our two species here, and we can collect animals from allopatry. And we rear them either with a single conspecific, member of their own species, or with a single heterocivic, the other species. And then we give them limited amounts of shrimp, and then we look at the production of carnivores in the lab. And what you can see is that just as we found in the natural populations, the plasticity here within a single generation basically will mediate that divergence between those two species, right? So what's happening is that the Balmofrons are just a little bit better at getting the shrimp Q, and that's why they're producing more carnivores. The important point, though, is that by basically using this plasticity, these two species are able to stably coexist with each other. Now, that divergence, which occurs within a generation, and we think became fixed within just a few dozen generations. And so as I said earlier, we have a number of lines of evidence to suggest that Balmofrons has expanded its range southward and westward into habitat previously occupied by Multiplicata, and only maybe fewer than 75 spadefoot generations. So there's the plasticity on the left there, the experiment that I just showed you. And what I'm going to overlay on top of that is the data from the wild that I showed you earlier. And you can see they basically sit right on top of each other. So the hypothesis here is that the plasticity in this system basically jump-started those canalized differences that we now see in natural populations. And in this way, we think plasticity-led evolution can really promote, again, rapid change. And the last point I want to make is that support for plasticity-led evolution is increasing. So I've talked about one example today. As I said, there are many really nice examples out there. Here are just a few other examples. At last count, there's about 40 or 50 pretty good examples, I think, in the literature. And so, it's not just something that we find in a few oddball cases, as some people have suggested. You find evidence for this in taxa ranging from plants to animals, invertebrates to vertebrates. But of course, more work needs to be done. So I want to just conclude by coming back to my central question, which was, what is plasticity's role in evolution? And as I hope you've seen today, by enabling populations to undergo rapid and often adaptive change in direct response to changing environments, plasticity really does add a new dimension to our view of evolution. And I want to say really quickly, it also adds a new dimension to our view of ecology, because without plasticity, species may not be able to coexist, for instance, as we saw in the spadefoot example. But with regard to evolution, we've seen how plasticity might jumpstart phenotypic evolution in an adaptive direction, with genetic changes evolving later. And this is why we can say that plasticity illustrates this idea about how phenotypic evolution is led by plasticity, and that genes are followers, not necessarily leaders, in adaptive evolution. But I want to just conclude by saying that it's not like we can all go home and do something else or whatever. There's much more research I think needs to be done to clarify plasticity's role in evolution, evaluating the buying time hypothesis in addition, but also evaluating the plasticity-led evolution hypothesis in diverse systems, looking at the conditions under which it occurs, using theoretical studies, using experimental evolution studies, but importantly also using more studies of natural populations to infer situations where it might occur and where it might not occur. And so with that, I'd just like to close with my acknowledgement slide to thank the people and the places, the funding agencies that have made this work possible. And also to say a really loud merci beaucoup for the invitation and for listening.
All right, thanks a lot. So we can't applaud, I guess, but very interesting talk, lots of questions. So let me start get the...
Actually, before we get to the questions, I just want to put in a plug really quickly.
Yes.
We have a book coming out on plasticity and evolution that addresses some of these issues. And I'll just leave this up. Here's the table of contents. I'm not trying to sell this book. In fact, I'm going to argue you shouldn't buy it. We're really fortunate we're able to get some money to make this book open access. So when it becomes available in early May, you can just go to the Taylor Francis website and download the book for free. And again, it's hopefully you find it of interest. So
great, yeah, that's the right audience to promote it anyway. So we had so people can vote for questions. You can keep voting for this question. Someone is saying this is a good book. This is not a question, but so we had a question by Sonia Sultan. So Sonia, I think your mic is on, so you can ask your question.
Great, thank you, Louis, and thank you, David, for a really deeply stimulating and intriguing talk. I wanted to pursue a little bit part of your hypothesis about the process of plasticity-led evolution, because I'm just curious, and I believe there's another question about this as well. I'm just curious about that step where you state that canalization will occur. I guess in your system, in your case study, the evidence for that is the RNA-seq data and the PCA that you showed, those overlapping and less overlapping ovals. Mm-hmm. But I just want to ask you about that, about where that prediction comes from exactly, that plasticity will be lost. I understand it may have happened in this case, but I just wonder why that's a general prediction.
Well, so I thank you for the question and for attending, Sonia. I don't think it's necessarily a general prediction, right? So one of the things that really confuses and puzzles me, and I'm sure some of you guys in this audience know a lot more about this than I do, is why genetic assimilation occurs, right? And so we don't expect genetic assimilation generally to occur. And so we see genetic assimilation seems to be potentially happening in this system, right? So they're losing the plasticity. And I say it's a mystery to me why that should occur, because it seems like, why not just maintain the ability to be flexible, you know? So in many cases, you probably aren't going to get that, right? We do know, though, in this particular situation, that if you, to go to your question here, if you take those animals from those populations and you're not going to get the omnivore phenotype being produced in them. You know, it's really hard. I mean, you get it in a few individuals, right? A few SIB shifts, but for most of them, they're just like, they're born as carnivores. So it looks like they've undergone that cannibalization and they've lost the diet-induced plasticity that was present in the ancestor.
Do you have a hypothesis why that would have happened in this case? Do you think there's some, is it some kind of genetic change, or do you think there's a cost that caused that?
So that's one of the things we're trying to look at now. I mean, we have a genome to work with. Took some time, but we're trying to figure out what's happening genetically. Whether we can ever get there, I don't know. But I think what's happened here is that selection is just really, really strong for these animals, this lineage, to only produce the carnivore, right? They're just not very effective at being omnivores, just as the other species not very effective at being the carnivore morph. And so it's just very, very strong selection. So, the character displacement story, which was, sort of preceded all this stuff, and that's all been published, it's basically documented that strong and consistent selection in these populations. Now, we don't think it's been operating for a long time, but again, we think that selection is really favored the production of only, the fixation at carnivore morph. And, and there's many reasons for why you would expect them to lose the plasticity, because, you're doing something very different to produce an omnivore than produce a carnivore. Morphologically, it's like they're opposites. Behaviorally, they're opposites, so there's all these, it's like you can't do both, right? And so, so once you have a situation where it's only that carnivore morph being favored, then they've lost that ability to produce that omnivore morph. But again, we don't know mechanistically why, and I'm not sure we know for sure why they completely lost that.
All right, thank you.
Okay, so I think we can move on to the next question. Thanks, Sonia. Andreas had a question, very short, but many people are interested in. So
it's a great question. I mean, any evidence for cost of plasticity in your system? And the short answer is no, not really. I mean, we've looked at it. We haven't looked at it as well as we probably should have, but we have not found any obvious costs in this system to the plasticity, which again sort of deepens this mystery as to why, why you get genetic assimilation, why you get the loss of plasticity.
The ancestor is plastic too, so
yes, and so
it doesn't express it in a
right. And many of y'all know, I'm sure, that costs of plasticity have been a really thorny problem to detect, and so this is not just restricted to our system. It's been a problem generally. People who study plasticity is detecting those costs. But yeah, we haven't detected any clear costs in our system. So Tom asked.
Sorry, Andreas, you're happy with the answer? Yes, I guess.
Well, any project what you would do in order to further verify for costs of plasticity in such a case?
You mean in this system? I mean, so, I guess, there are many people, again, there have been a number of publications that have come out that talked about, ways to consider evaluating costs. So obviously, doing things in natural setting as possible, using multiple environments, things like that. That's something we probably should do. We just haven't gotten around to it. So, but yeah, it'd be great to figure out if we have those costs here.
Okay, thanks.
Yeah.
All right. So Tom,
Tom asked, is there any evidence of transgenerational plasticity in the polyphenism? Yes, there is. So the story is a little bit more complicated than the way I presented it, in that the multiplicata, and sorry to use all these scientific names, but the one species, multiplicata, which is the species that I didn't focus on so much today, the character space in that species seems to be mediated by a maternal effect. And so we think this is the species that has come into contact with the other species probably really, really recently. And so what we find is that basically by being forced to produce only omnivores, to tell you about the scenario here, they mature as smaller adults, and those smaller adults seem to be predisposed to producing mostly omnivores in the next generation. So they produce smaller eggs. The smaller eggs hatch out as smaller tadpoles. The smaller tadpoles are more likely to be omnivores. And so you get this sort of self-reinforcing cycle going that's mediated through this condition-dependent maternal effect that we think is explained in part the sort of channelization towards the omnivore morph in the one species, right? So this is the opposite species than, again, what I talked about today. And so we think that transgenerational plasticity has probably played an important role, again, in driving that. So I actually have a graduate student who's studying this right now to try to take this apart. But one way we like to think about this is that, you know, the transgenerational plasticity may be acting as a bridge between the sort of within generation plasticity on the one hand that sorts out these two different ecological niches, and then perhaps the sort of more genetic canalization that we might be seeing, for instance, in Spiabomophons and the other species, which might take longer to evolve, but that the transgenerational plasticity could basically serve as the bridge between those two. And that work has been published. So that was in Evolution in 2009, 2010. So if you want to read more about the transgenerational plasticity, it's in there. Did that answer your question, Tom?
Yeah, perfect. Thanks, David.
All right. Those who asked their question, can you please turn off your microphone afterwards? So there's an anonymous person in the audience who asked several questions. So I don't know the name. I don't know how to turn on their microphone. So you can just read that question about the mechanisms, whether you know about anything involving. Yeah. So we're trying to figure out more about what's going on with the epigenetic mechanisms. We've got a great collaborator just a few miles from us here at UNC, a collaborator at Duke, so Jenny Tung, who's you know, done a lot of epigenetic stuff in like baboons and stuff like that. And so we've been working with her, and we have some preliminary data, but nothing really definitive yet. You know, so you know, we found some methylation differences. So most of this is looking at methylation, I should say. And so we're finding differences in methylation patterns between carnivores and omnivores. That's not super surprising. We're also finding some differences, you know, between some of our populations, but it's been slow going. And so we hope to have more information on that down the line. But that's an obvious place to look. Yeah. All right. So you have many questions. I don't know if you'll be willing to answer to all of this, because this has been a very successful talk. So lots of people. So perhaps we'll switch to the next person, Anton. So Anton's a modeler, I believe, theoretician. So Anton, can you?
Thank you. Yes. So actually, I think this topic itself is very important and deserves much more attention from the community in biology and other sciences. But my question is the following. I wonder, so you showed that you studied the plastic response of the population. You studied the uncovering of the cryptic genetic variation. I wonder if you have any data in this system that can say something about isogenic variation. So you take, I don't know, maybe inbred line or close siblings and look at the errors in development. So I wonder if those, so like abnormal development results, yeah. I wonder if those really somehow mirror the morphs, phenotypic morphs that you observed during this evolutionary process that you described. Yeah, no, that's a great question, Anton. Thanks for asking. We haven't done that. We're actually trying to do that now. We now have a situation where we can rear these guys up, you know, create these isogenic lines, and we're actually doing that right now. I mean, one of the bottlenecks for us has always been taking care of the little
little baby toads, because they take a lot of attention. And so now we do have a crew that's doing that. And so we hope to be able to answer that question. But that's actually a very interesting question is, you know, yeah, do you get these abnormalities and can you see, you know, vestiges of these like this carnivore mark being produced by some of those abnormalities? And that's definitely something we want to look at. Again, unfortunately, I can't give you a definitive answer right now about what we've seen because we haven't done that yet, but I think it's a great point.
If I might say something, I think that you might look at fluctuating asymmetry in your animals just to measure developmental stability and the occurrence of developmental noise.
Yeah, great idea. We have done that, and you know, we find more asymmetry in carnivores, you know, than you do in omnivores, and that has made sense to us because, you know, it's the you know, the omnivore is sort of developmentally derived and, you know, evolutionarily. But yeah, you do find greater degrees of fluctuating asymmetry in carnivores than omnivores.
Vincent, I think you had a question yourself that you're keeping.
Oh, yes, that's right. So I was wondering if you had any data on the adults and whether whether this plasticity extends to other phenotypic traits later in life, because it might be interesting to investigate the genetic correlation and maybe some trade-off between the selection during the early phases and later in life.
Yeah, so the limited data we have on that, Vincent, and again, great question, is that there doesn't seem to be long-term differences, right? So we published a paper last year before I came over, but in the Biological Journal of Man Society, and you can download this from my website as a PDF, where we collected carnivores and omnivores from the wild, and then we reared them under common conditions of sexual maturity. And we did not find differences in survival or you know, body size at sexual maturity and things like that. I think the important caveat though to that is that we reared them under sort of common idealized conditions in the lab, and these guys are almost certainly not experiencing, you know, identical conditions in the wild because, for instance, carnivores get out of the ponds first. Many of the carnivores are already out of the pond before the omnivores even begin to get out of the pond, even before they metamorphose. So, you know, they're experiencing a different environment. I mean, you know, the environment's different when they first come out. We don't know much about what they do in the wild. We wish we knew more. You know, like, do carnivores disperse farther, or do they disperse shorter distances? Do they have different habitat preferences and stuff like that? And so, you know, we'd love to know answers to those questions to get at whether or not there are long-term, you know, long-term life differences between the morphs. You know, so most of what we've had to do is just basically, like I said, rear them under common conditions.
So at this point, I think you can just choose the questions because you won't be able to answer everything, I guess.
So I just the one I'm seeing right in front of me. So Christian asks a great question again. What's the difference between plasticity first and plasticity-led evolution? In my mind, they're the same thing. So we initially, and I don't want to claim credit for these terms. I mean, I'm sure somebody else coined them somewhere along the line, but you know, when we published our tree paper, we used this phrase, plasticity first evolution. And you know, people were pretty critical of that term because I think they, you know, just as they've been critical of Mary Jane's genes as followers and not necessarily leaders in adaptive evolution, I think many people thought we were trying to say that, you know, genes played no role here, that plasticity came first. And so the arguments I would get, people would say, well, obviously you have to have genes in order to have plasticity. And it's like, well, yeah, you know, and so I realized that, you know, a lot of times, you know, in science, just like in, you know, outside of science too, in politics, right, we get into arguments because we're not talking about the same thing. And so, you know, we made this change to just start calling it plasticity-led evolution and just expunge the plasticity first out of our vocabulary so that you wouldn't basically confuse people. And again, as I said, when I was giving talks and stuff, I get a lot of people would be confused about what we meant by plasticity first. So that's basically all it is just to make it a little bit more accurate, I think.
So there's another kind of conceptual question about the definition of novelty. I'm a bit curious about this.
Yeah, so obviously, and many of you guys know this, I mean, novelty is a super hard thing to define, right? And you know, and again, I've had, you know, mentioned Doug Fatuma earlier, you know, he's maintained that, you know, this is not an example of true novelty. You know, this carnivore morph, you know, I'm not gonna go to the wall to argue with him on that, I suppose, but you know, I guess you get in a problem of like, well, what is truly novel, you know? And so, you know, one of the other things I've studied is the origins of the rattlesnake rattle. And it seems like, oh, that seems like a novel structure to me, you know, because there's no sort of homologue that you find. But, you know, a lot of these cases where you have a novel complex trait, you're probably not going to have true novelty, right? Because if it's a complex trait, it's going to have vestiges of pre-existing, you know, traits that are going to be sort of tagged along that.
Well, the question came from a philosopher. So Antonine, you can specify what you meant, perhaps?
Yes.
I don't know if I can describe the philosopher. Is that the right description?
Probably. I'm teaching philosophy. I prefer probably to say that instead of I'm a philosopher, I'm also doing sometimes history. So yes, my question was actually about your definition. I know that you tried a little bit to avoid to answer this question of novelty, but I was wondering, since you quoted Mayer, if the idea that you had about novelty was something close to a working definition of Ernst Mayr about novelty, that he described as something which was something functional, or if you had something more precise in mind, or
I don't, honestly, I don't have anything. I'm not a novelty expert. I think that pretty much captures, I think, my view on it is Meyer's idea that, you know, it's novel. This is you know, if you look at this phenotype, you know, if you characterize it morphologically, it's like way out there, you know, like if you do a sort of multivariate space. If you look at it behaviorally, it's like very different. And the video didn't really do justice to it, but, you know, ecologically, these guys are doing something radically different ecologically than all the other members of this clade do, you know, in terms of utilizing this distinct diet. We think that, you know, the origins of this morph probably explains in part why they're able to invade this desert environment. So these are all sort of functional definitions, components of that novelty thing. But, you know, you're quite right that novelty is a thorny thing. And obviously I started my talk off with saying, you know, I'm going to talk about the origins of novelty. And so maybe I should be more precise in my definitions, but.
No, it's also because you mentioned this, at some point you mentioned this spike coming in the mouth. Yes, and I was wondering, because you didn't say if you have any idea. I mean, you say so. You say that.
So we don't see anything like that in any of the other lineages that we studied. And then when you first start seeing it, you start seeing this little like black keratinized spot on the upper mouth, and then it just sort of like that spot starts to grow into like this, you know, pyramid, and then it starts to grow, you know, that pyramid becomes elongated. So we just, you know, found this a couple of years ago, and we want to know more about like what's going on there. But so yeah, it's not like it's not obviously growing from something else. It just seems to be there, you know, and so that's partly why I refer to it as a novel trait.
And it's not necessary for eating, is it?
It's not necessary for eating, because you know, the omnivores can eat shrimp. They're not super effective at it, and they're able to deal with it. But obviously we should, you know, one of the things we want to do is just to, you know, file it down on some individuals and just see what happens to their feeding abilities. And I'm guessing they just won't be as good at getting the really large struggling prey. And I sort of passed over this, but these guys, as they get bigger, they become really cannibalistic, and so they start eating. They shift from eating mostly shrimp to eating a lot more tadpoles, and so they might be using that spike more for really large prey. They don't have hands, obviously, so they can't grab onto things. So it's basically like using that beak and presumably the spike to impale things and to pull it in. And so that's probably what they're using it for.
Okay, thank you.
Thank you. So I don't know if we don't want to take too much of your time. It's true that we like discussion in these meetings because we don't have them very often, and it's nice to have. But I think, so I want to tell people I removed some questions that were similar to other ones. So I guess you can pick up one last questions from this list.
Okay, so anonymous attendee says, it looks like the variability of the fitness is also higher in the carnivore type toward the right of the graph. Could there be a reason for this heterogeneity of the variability? And so, yes, that's quite right. So with our fitness proxy being body size, carnivores are typically larger than omnivores. And so what the questioner is referring to is when I showed you that slide towards the beginning about the disruptive selection, it was not sort of symmetrical. The node on the right, which was associated with more carnivore-like individuals, was actually higher. So we think that carnivores are actually, if you just look at it like this, that carnivores actually, they're bigger, and so they should generally do better. But we think carnivores, we find that the competition among carnivores with other carnivores is much stronger than the competition among omnivores with omnivores. Because omnivores are generalists, right? And so when they are competing for, say, detritus, they can just, an individual can shift to eating like plants or bacteria or maybe an occasional crustacean. Whereas the carnivores are basically gone all out for the meat. And so they're competing only for those shrimp or maybe, like I said, other tadpoles. So we think they pay a real cost to that specialization. So even though they seemingly do better, overall, we think they probably are about equal with omnivores, essentially. And we see that, with doing the frequency dependent effects. And so I think that's, partly the answer to that question, is just this cost of specialization.
All right, so questions keep coming. I don't really know, perhaps people can write to you afterwards or.
Yeah, sure. Feel free to write to me. I'm happy to.
Yeah, because I think we've been there for quite some time. So I mean, if people can write to David or write to us, and we'll write to him. So I'd like to thank you again for a really great talk. You've seen it was very inspiring for our audience. Questions keep coming. I would have had questions probably, but I can write to you also. So thanks again, and so this talk will be broadcasting on YouTube shortly, and then we'll let you know when we have a next webinar of this kind. So thanks again, David.
Yeah, thank you guys. Thank you, everybody.
Thanks a lot, David. Really great talk. Yeah,
Thank
really
you.
nice talking to you.
Thank you very much.
Thanks a lot.
Bye.
Hey, David.